Hello, everyone, and welcome to the OmicsLogic training program and course on the analysis of SARS-CoV-2 genomic data. We were scheduled to start the program from June 8th, but due to some natural calamity, we had to reschedule. So I would like to welcome you all once again. This is a very unique program designed by experts to help students, faculty, and researchers looking for ways to understand the virus in greater depth, to be able to find ways to minimize the impact. And the impact of 2020 coronavirus pandemic or 2019 coronavirus pandemic has been unimaginable. So, without wasting much time, I would like to start talking about some of the concepts, and I hope that many of you have joined. And we are recording this session, so if anyone has missed, you would be able to watch the video later. So, COVID-19 stands for coronavirus disease, and 19 is the year this pandemic started. The pathogen causing this disease is SARS-CoV-2. That stands for Severe Acute Respiratory Syndrome Coronavirus 2. So in this program, we will be learning about the genomic data of coronavirus, and also we will be comparing the viral genome to other viruses. Further, we would do the research together by learning where to find the data and how to analyze it. So, as I was saying, by analyzing publicly available data, you will be able to see for yourself the clues into the potential origin of this novel coronavirus and understand how the variation between this virus and other similar coronaviruses might affect its biology, cell entry, replication, or maybe transmission. So in this program, we will learn about genomic data, right? So this program will be held completely online in regular online sessions like this one, supplemented by independent online learning and practical assignments you can complete on your own schedule. The program is aimed at beginners, including high school students and non-bioinformaticians that are interested to see how genomic data in general and SARS-CoV-2 related genomic data can be analyzed to study the viral pathogen. So to make this program, accessible and allow for seamless analysis, interpretation, and visualization of data, we will rely on cloud tools made available on the TBioInfo platform. So all the participants of this program will have access to the TBioInfo platform that has dedicated sections for analysis of genomic mutations, evolutionary analysis of whole genomes, phylogenetic analysis, and characterization of various data types in the context of viral host interaction. But before we start the actual topic, I would like to give you a quick introduction about myself and our company and our goals. For those joining us for the first time, my name is Mohit Mazumdar, and I have a PhD in computational biology and machine learning, and I'm one of the global representatives here in Pine Biotech. Our vision is to enhance human health and well-being by enabling biological research and discovery with relevant data, solution, and support. Our mission is to simplify bioinformatics uh, and advance research through our modular and intuitive multi-omics platform powered by human intelligence and human experience and artificial intelligence. And around the year, we work with students and researchers and provide research support and big data analytics tool designed for biomedical data discovery. So one second, I'll just look into, thank you, Bipsha, for addressing those comments. So as I was telling you that I'm honored, actually to be working with a great team here that has a diverse experience in various areas of research, including our CEO and co-founder Ilya Brodsky and other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background, and has expertise in oncology, immunology, and biochemistry of inflammation. So he has a long-standing recognition for his generous support for many international projects and research activities. One of these projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonard Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics, and over the course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. This has been widely used in basic and translational research in biotechnology, pharmaceutical research and development, agriculture, and biomedical research in general. What we will discuss, what we are discussing, or what we will discuss today is also a hard work of people mentioned here in the left. Julia Panov is an expert bioinformatician and helping us preparing many of the materials that we'll be using today. Avi is a computer scientist and leads the development of T-Bioinfo platform that we will be using. Joshua maintains the educational web portal we will be leveraging, and Deepsha, our community manager and support specialist, who I'm pretty sure many of you have interacted with. So many of you joined us today, and we have been posting about the program through the landing page. Many of you have joined today who are through the LBRN University Network. So the way to join the program, I just wanted to again discuss because to simplify this, the first process of registration, as you know, starts with filling a form. Then the form redirects you to the to the organization page where we will be where we register you on the platform, on the educational platform, where you have a profile. So we will be discussing all those things in much more detail in today's session, so that none of you have any issues regarding that. So please feel free to ask questions, and we will be addressing them at particular time points where we will pause and see that if you are facing any problem, you have any questions regarding that, and we will address those questions right now. So as I was telling you that being part of these organization pages gives you access to a lot of resources. So one of the organization page is the one that you see in the screen. Another organization page is the LBRN organization page. So let me go there and show you what are the resources that you have, and how can you utilize those resources to learn the most. So this is the first organization page through the online registration. So in this organization page, this is the link. So let me share it with you all. Okay, Bipsha, thank you. You have already shared, I think. And also there is another organization page for the university account, and this is the link. It's the page with similar features or same features.
Yeah, I've pasted it already.
Okay, thank you, Bipsha. So let me show you one example, because both of them are actually the same. They both have same features. So it's just that people are registered into two different pages. So here, this is the home page, and here what you will see is all the upcoming events that are associated with this program. So today is 9th because it was already scheduled, so it shows 8. So today is 9th, and we have this first session, and our next session is on Friday, 12th June. So you can see the sessions when they will be the upcoming sessions in this event. And here below, you will see the organization updates, which means that all of the participants, all of you here, will be able to see whatever the other participants have been doing. So you'll be seeing the updates, that if someone has finished a pipeline, if someone has finished a course. We will go into much detail after in a while. So second feature is one of the most important feature, and that is the forum. So let me click here. So forum is an interactive place, and it's a place where you will find answers to your question. You will find all the resources, and you will also find the video recordings and whatever we do during the sessions. So here you can see that people have already started a topic, like Shreya here. Thank you, Shreya. And Sumya, she also has started a topic. So here you will see session recordings. So when you go inside this topic, you will find the recording of this session and the upcoming sessions as they go on. And also you will find here session data files and presentations, which means that the presentations that we are doing, or the files that are associated. For example, today we shared a reference publication with you, and in the upcoming sessions we will be sharing data files with you that we will be using for the data analysis. So all those things will be available to you all the time here in the forum. And in the forum, you can also start your own topic, asking your own questions, that whatever problem that you have faced while running some analysis or doing a course. So you can write it here. This is the place to do it. So as I was telling you that there are several resources associated with this course. So let me go back uh, to the presentation. So before that, I just wanted to show you the syllabus also. So you are free to explore this organization page, and I would advise you to do that. So in the syllabus, you would see detail of each session and the associated material with each of these sessions. So we have online courses that that explains many of the things that we will be discussing today. So how you can benefit it with something like an additional resource that you can go on and read and understand a deeper and develop a deeper understanding of the subject. So the first session, as you see, is associated with Transcriptomics 1. So here there's a project in the second session. So you will find all the details of the sessions here, detailed sessions in the syllabus tab. And the courses are the courses that have been associated with this program, and the projects that are associated with this program. So let me go back to the presentation. So as I was telling you, that you would find all the information, relevant information in this page, in your individual organization pages. And you have to make sure that you have access to both of the platforms, because in this interactive program, we will be discussing and we will be doing analytics, and we will be referring to these sites all the time. So make sure that you have the login information with you, and you have been able to log in into the server. And if you have any issues, please reach out to us, and we'll be helping you with that. So these are some of the courses that I was talking about. Courses and projects that we will be discussing during the sessions, and it would be nice for you to go in more depth and to be able to understand and practice a little bit on your own. So one of the very important thing about this program, like the OmicsLogic SARS-CoV-2 program, is that that each of you will have access to your own personal dashboard on the educational platform. So a dashboard or a profile with a complete name and a good picture would give you access to a wide range of people who are there in this community with diverse skill sets. They would be able to see your profile, and that name would reflect on your certificates as well. So it is very important to have a good profile picture and a good and your complete name, so that you will be able to access and see these use these certificates. And these certificates will be there in this profile along with you all the time. So you can share this link and associate this link to your professional accounts like LinkedIn, so that the certificate will be redirected to this page, and you, anyone who is interested in learning about you, can see the certificates online as well. So one of the reasons for this, and also to talk about the outcome of programs such as this, is with the right dedication and focus, this program will help you develop an independent research project and apply those skill sets and concepts you acquire in a data analysis challenge. So Olga here, she is our previous participant from OmicsLogic precision medicine program, and she completed her training and then worked on a unique noble project on cancer. So after completing this training, you will be more confident and experienced in multi-omics data analysis. So how to reach there and how to achieve that is something that we will go through in this entire 1 month program. But here you can see a batch associated with her. So we have coming to that. So this unique OmicsLogic academic program is designed in a way that helps us communicate with each other and create many bioinformatics research groups within this program. So this is done through a tracking system. So where all the participants, and I was showing you that in the organization page, so where all the participants and the mentors are allowed to see other participants' activity. And it's a great place to track your own personal progress as well. So what happens is that once you complete a course, you get 500 points. Once you run a pipeline, you get 100 points. Once you update your profile, you get 100 points. And once you do the group activities like commenting on the forum, commenting on someone else's activity, favoriting or liking that activity, would give you also small 10 points. So too, I mean, in the end, I mean, when you are doing this analysis, when you're utilizing the resources, you will gradually get into those, become such profile, and you will be shown in the leaderboard. So that is one of the benefits. So if you have any questions, like before we jump into today's topic, the actual topic, let me ask you that if you have any questions regarding the program schedule, resources, and registration, you can unmute yourself and ask me, or you can write it in the chat as well.
Okay, thank you, Mohit. Maybe in the
Okay.
meantime, we can switch the screens.
Yes. Okay. I'll pass it on to Ilya now for the rest of the presentation.
Okay, thank you, everyone. And thanks, Mohit, for walking us through the registration and the different resources that we have in this program. Again, like Mohit mentioned, you can post any questions as I'm going through the topic for today, and he will be responding to any questions about registration, access to the online platform or to the educational portal, and making sure that you know how this program is organized. So today's session is going to be a little introduction into sequencing viral genomes, and specifically into the technology of next generation sequencing. So the technology has really developed over time. And the first thing that we'll talk about is kind of a little history of how we got to where we are today. What type of available data are we going to be going through in this program? And we'll give you a little sense of how the program is going to work over the next month or so. So a little history of genomic data. As you all know, in the 1950s, the structure and function of DNA was discovered. And of course, the Human Genome Project is one of the first achievements that were made possible as a result of sequencing. And by 2006, Illumina has launched introducing this new shotgun technology that is now making possible thousands of genomes and an accumulation of next generation sequencing data that really is accelerating, as we can see in this chart. So what type of information can we get from next generation sequencing? Next generation sequencing is one type of omics or multiple types of omics data that characterize different subcellular processes. So when we talk about subcellular processes, we are going to discuss phenomics, how cells look, genomics, what is in the DNA and variation in the DNA, epigenetics or epigenomics, popular, a more and more interesting and popular technique that allows us to study variation that is not based on what is encoded in the DNA, and transcriptomics, or how different genes are expressed. And as you can see, all of this is related to human omics data, right? So what is happening in the human cell, in the human DNA, at the human epigenomic variation, and also expression of human genes. So how does that relate to viruses? Well, the viruses are a virome, and they really work in communities that are present in inside of us and around us, and also in other animals and other species, including infecting bacteria. So the virome is a part of the microbiome, or a collection of microbes, and it constitutes all of the organisms that inhabit us, and the different types of bacteria and viruses coexist in a variety of environments. Studies of the actual virome as a whole are really novel, and are, have started to become more and more popular over the past couple of decades. But as you know, similar to bacteria, before, when we wanted to study a virus, that virus had to be isolated and studied after culturing that in a lab. And so the virome is really an attempt to study the viruses in their natural environments, and those viruses coexist as communities. The size of the mammalian virome is actually not known, although our own cells are outnumbered about 10-fold by our bacterial microbiome. The number of viruses may be 10-fold higher. So it's a huge diversity and number of different viruses. Human feces alone contain about over 100 viruses per gram. And also the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. So it's a really fascinating situation. And in fact, viruses play a variety of roles. Not all viruses cause disease. Many viruses actually help maintain a balance between the different types of bacteria that are present in the gut. And not all viruses actually can enter into the human cell. So we coexist with viruses in many different environments, and only some viruses actually cause this disease. A lot of diseases are normal and cause only mild symptoms, but some diseases, like SARS-CoV-2, cause this abnormal pathogenesis. And one of the questions that we'll be exploring in this program is how the virus could have originated, where could it have originated, how could it have first entered the human body, and how does the transmission happen, and what exactly does it do at the biological level. And so one of the technologies that allows us to study in very great detail how viruses enter the host, how they replicate, how they cause disease, is this technology called next generation sequencing. So next generation sequencing, specifically for viruses, but for any kind of sample really, what happens is you have a biological sample, and then that biological sample contains DNA or RNA, and that DNA and RNA has to be converted into digital data, which then has to be analyzed. And so today we'll talk a little bit more about that process of extracting a sample and eventually turning it into this digital sequencing data. So metagenomic and viral sequencing could be of several different types. For example, targeted sequencing, which means that specific sequences are amplified. And by looking at those amplified sequences, we can search for specific viruses. It's a lot similar to real-time PCR, where we are only looking for a specific strain, or we're looking for a specific family of viruses. There's also a possibility of detecting novel viruses in total DNA or RNA of the host. So if we don't do any kind of depletion of that DNA or RNA, we're able to detect presence or even sometimes quantify how much viral DNA or RNA might be present. And there's a lot of challenges associated with doing that to really differentiate between what is and what is not a true viral RNA or DNA. And then there's amplicon or environmental, where we attempt to sample everything that is in the environment and focus on a specific type of sequencing. For example, there are primers that we can use to amplify specific sequences that are only present in viruses or only present in bacteria. For example, in bacteria, the 16S ribosomal RNA allows us to focus on only bacteria, or even hypervariable regions of the 16S ribosomal RNA allows us to study specific variation inside a collection of different bacteria. So a number of different sequencing techniques. Those techniques have been evolving over time and have been originally used only for research purposes, but in recent outbreaks, those have been already started to use in a clinical sense. And in the clinical sense, it all started around the time of the recent outbreak of the Ebola virus in 2014-2016 outbreak in West Africa. During that time, groups of World Health Organizations and other types of health organizations were sequencing on the spot genomes that were extracted from patients. And you can see here a couple of examples where typically you have a clinical specimen, you use specific types of primers to amplify those sequences and map those sequences onto known viral genomes, for example, databases of viral genomes, or potentially look for specific types of sequences that align to databases of known genome or of known genes, of viral genes, and an ability to also eliminate host genes from whatever we find. And so once we find those non-host reads that are present in a sample, we can then analyze those for what types of viruses and how the virus genome is covered. Also, in terms of clinical metagenomics, that there has been an interest in also transcriptomics, right? So one type of sequencing is to detect the DNA that is present or the RNA of the genome, because some viruses are RNA viruses like the SARS-CoV-2. But sometimes we're also interested to understand what types of genes are being produced from that genome. One of the resources that we have made available to understand some of these techniques, like we mentioned, are applied both to viruses and to bacteria. It's a similar process, but to really understand some of these details, we have a number of resources that you'll be looking online, and I'll talk about this a little bit further, that you can complete to kind of get some more background information on this type of sequencing. The basic idea is, and the innovation behind this, is that really we can study viruses as they appear in their natural environment. So we can sample them, we can then quantify how many reads align to a specific genome and study that genome. Now, to talk about this type of sequencing, we have to talk a little bit more about the different data formats that we'll be using throughout this program. So there are going to be three basic types of formats. FASTQ files, which contain individual reads. And so you have to understand what a FASTQ file looks like to really know what the process of analysis is required to extract meaningful information out of this kind of collection of short reads. There's a FASTA file, and that FASTA file is typically used to contain sequences of genomes or individual genes or other genomic elements. And then there's typically a GTF file, a genome transfer format file, that contains information about the beginning and the end of specific genomic regions. So remember some of these data types, remember that we will be utilizing them, and as we get more and more into the analysis part, understanding of these different genomes is going, of these different file formats is going to be important. Okay, so now we have some interesting questions. How the platform is compared to using Terminal and other tools such as Minimap2 and SAM tools. Technical question, right? What is the output of next generation sequencing data? I will be talking about that in just a second. And can we identify mycoplasmas through metagenomics? So all great questions, right? I suggest looking at some of these online resources. That's why we provide them, is for filling in the gaps as we go through the session. Today's topic really is to focus on viral sequencing and explain how the sequencing technology really conceptually, what information does it contain that we can analyze. So let's take a look. When we are talking about SARS-CoV-2, the original genome from SARS-CoV-2 did not come out as a sequence of nucleotides already assembled. So when we first see the output of next generation sequencing, it looks like short reads. Each individual bar that you see on the screen represents a read. And that read is a maybe anywhere between 30 and 300 base pairs long sequence, nucleotide sequence, containing letters A, T, C, and G. And the sequence can be aligned to some reference genome. So here you can see beta coronavirus Wuhan. WEAV stands for Wuhan Institute of Virology, 04/2019-2. And here another line, you can see the consensus, and this is coverage. So what happens is after you get those short reads out of the next generation sequencer, those reads need to be extracted from a FASTQ file or from an SRA file and mapped onto a reference genome. Now that reference doesn't have to be perfectly containing the same sequences. It could have a variety of different regions that maybe don't have a very good mapping quality. Nevertheless, what that allows us to do is to understand the overall sequence of this genome and create what's called a consensus. The consensus is really where each individual read is agreeable to what is in some kind of a reference, and also agreeable in terms of the differences from the reference genome. So here you can see the result of that analysis after that those reads are aligned to the reference genome. And you can see here one of the FASTA files that contains the full sequence of this genome. So what does it contain? It's an accession number, and then there will be a description of what exactly is in the FASTA file. So here we see severe acute respiratory syndrome coronavirus 2, isolate 2019, novel coronavirus USA, WA stands for Washington, 2020 or 1/2020. So this is January 2020, complete genome. Right, so this is the consensus, right? Just to have a sense of the difference between the FASTQ file, which contains all the individual reads, and a FASTA file now, which is the consensus genome. Those consensus genomes can have differences. For example, here you can see that these two, this is a complete genome, and this is a complete genome, and this is the beginning of that genome. They don't start from the same sequence. Okay. So those types of differences could be studied then to compare different types of genomes. What do the peaks in the coverage region signify? So the peaks are associated with the number of reads. Coverage is the number of reads that is aligned to that particular position. So if that coverage is higher, you'll see that that means more reads aligned to this region. If the coverage is much smaller, maybe there's not a lot of reads that are aligned to that. What does it mean in terms of reliability of the sequence that we see in the consensus genome? That tells us that wherever we have high coverage, and that high coverage produces a consensus that is almost perfectly aligned between all the different sequences, that is a high confidence region. When we have a lot of variation, we either have less confidence in a specific portion of the genome, or potentially we'll talk about additional types of analyses that can extract other useful information from those different regions. Okay, so the novel coronavirus genome is the RNA of this virus. This is a positive strand RNA virus, about 30,000 base pairs long, so you can see it right here. The 30,000 mark for a virus is very long. In fact, it's the longest RNA genome of viruses. And so as we look at the specific genomes and as we look at the specific sequences, now we can study their variation. In the FASTA file, just so that everybody is clear, right, we have this continuous sequence of an individual genome or a portion of that genome. Not every genome is a complete genome. Some genomes that you will find in a variety of publicly available databases are partial. In the FASTQ file, you have those individual reads, and those individual reads will also have an ID, and then they will have a length, and the sequence of that length. In addition with that, you'll have a quality of the reading coming out from that sequencer. So after those reads are coming out, those reads have to be aligned to the reference genome, and this is kind of what it looks like. You have some kind of a reference, and each read is aligned to the best position on the reference. Sometimes there is a gap that is present in the read, and that will be denoted by these dashes. And sometimes you will also have some specific variation. That variation is what we call a mutation. Now, this type of information, the intermediate file of alignment between when it came out from the FASTQ file and now is being aligned to the reference genome, is going to be stored in a SAM or a BAM file. There are multiple ways that you can look at these, but the basic idea is that you have the beginning and the end of each one of your reads and any kind of variation or information that you have about that read and the read quality. For example, if we take some reads from a FASTA file and we align them to a reference genome, you will see that these red bars or the blue bars or the black bars represent individual reads. Here you have coverage. And in this example, you only see one region that is amplified. And in other regions, there's almost no reads, or those reads might be sort of randomly assigned to those places. So are there any questions about some of the basic file formats that we'll be using and about next generation sequencing and how we will be using that information? Because there's so much publicly available genomic data, and so we have to understand what type of data that is as we are going to start looking at it. So are there any other questions about this? I see a great discussion here about different aspects of next generation sequencing. So as you have any other questions, please add them in the chat. What's unique about this specific outbreak is that it really has produced a tremendous amount of publicly available data that is shared almost in real time. So back in April, we did a little analysis of the number of full genomes present in the human host that is being published per month. And what we discovered is that at least 4 times as many genomes, specifically in this outbreak, have been sequenced and made available as soon as those data sets were collected. And so I think it really shows us how accelerated the process of sequencing has become. The technologies have become much more accessible and affordable. And so what we did is we assembled a number of these genomic resources for virology research and education, and have a blog post where a lot of these different resources, places where you can access these genomic data sets and different tools for analysis of these genomic data sets in this blog post. And maybe Deepcha or Mohit, you guys can paste that link in the chat as well. Okay, so can consensus genome be a reference genome? Well, a reference genome is going to become a reference genome after it is validated. There are a number of different technologies that are used to really validate. As we talked about the way that next generation sequencing has evolved, that also made a number of different technologies compete with each other. And they provide different types of information because of the way that they generate those sequences. For example, there are techniques that make long reads, and those long reads could be anywhere up to 5,000 base pairs long, or today even longer. Nanopore, Oxford Nanopore, PacBio produce those long reads. Some of those technologies, like Oxford Nanopore, can contain a lot of mistakes along the way. And so it's useful that they are very long, but it's not as useful because each position is less reliable. But a combination of these types of technologies, including the traditional real-time PCR, RT-PCR techniques to really determine the full sequence of a genome, are used to produce a reference genome. But approximately what we showed here with the consensus genome is exactly what will be the outcome, right? A sequence, a complete sequence of a full genome, including some of the information about what's in that genome. For example, what genes and what other elements, genomic elements, are present. Okay, what is the difference between whole genome sequence and complete genome sequence? Well, whole genome sequence is next generation sequencing of reads from the whole genome, not just a part of the genome, but from across the whole genome. For example, what we saw in the previous slide, right here, this is called whole genome sequencing. And you can see that the reads are spread out across the whole genome. What is a whole genome that is submitted, for example, if you go to NCBI and you see complete genome? This is just a full sequence. It does not include the information about how many reads aligned to each position and what was in each individual read. Okay. Okay, so about in-depth information, like we said, there are a lot of resources that we cover specific to SARS-CoV-2 in this post and specific to virology. There are also general techniques about next generation sequencing and how that sequencing is done, and that will be found in some of the introductory courses, including Introduction to Metagenomics and Metagenomics 1, Transcriptomics 1, Introduction to Genomics. So we have a collection of courses where you can learn more in detail about the sequencing process, alternative technologies, and how this data is generated. Okay. What type of information is available as we start looking at all of these publicly available genomes? We can start analyzing this data to understand differences between different genomes as they spread across the globe or within a country, for example. And an excellent way to look at it visually is to go to this link. This is called NextStrain, and is a website that allows us to look at specific genomes as they went from the place where they originated and all the way to where we find them today. So here, if you go to that link, and there's a lot of variation that you can do in the site itself. So here you can click on play, and you'll see how this original outbreak originated in China, started spreading across Asia, and eventually in Europe, and we will see here around the world. And what you see on this side is how each individual genome from that location could be placed in what's called a phylogenetic tree. So we'll talk a lot more about these types of analyses, right? This is based on an analysis that somebody did. And each individual dot here represents one of those genomic sequences. So we can study the relationships between these different genomes, and then we can also look at variation within the sequence of this genome. And you can see this is a graph of diversity. So you can see how specific points in the genomic sequence start becoming more and more diverse. So essentially what we are observing here is diversity growing over time between the different strains. And as that diversity grows, we spot that diversity in only specific positions. It's not equally distributed throughout the whole genome. So this is just a useful visualization tool for us to understand how all of this type of information is linked together and start looking into greater detail at the genome of this novel SARS coronavirus. This site also contains all of the data, and you can actually download all of the data, including the individual genomic sequences that we will be analyzing in this program as well. Okay, so are there any questions about the different sequencing types or about the genomic data formats and about how they could be used to study this novel coronavirus outbreak and understand how the different viral strains evolved over time? As per graph, entire genomic sequence is different in different locations in the world. Yes, so that's exactly right. What we see is a different strains evolving over time and being more common in a specific location. And so we can take different locations and we can start looking at the differences between locations. So does the genome of COVID depend on location? Most likely it depends on the people because it spreads between people and people live in different locations. But the viruses, specifically RNA viruses, we'll talk a lot more about the process of accumulating mutations and those variations being as a result of a variety of different processes. But yes, those mutations accumulate and they become more common to groups of people. So groups of people could be, once everybody stopped traveling, right, those will be contained. And so we'll see specific mutations common to that group of people appear in different locations. What is the purpose of using different sequencing format? Sequencing maybe technologies, right? They evolved over time, just like any technology. Think of video technologies. We had videotape, and then we had CDs and then DVDs, and then Blu-ray, and then today we don't even know what those technologies are many times. So what is the reason for these technologies to evolve? There's always going to be a balance between cost, accessibility, reliability, and quality. And so when we started sequencing the human genome, that was the Sanger technology. Sanger technology requires a lot of manual annotation from which part of the genome does the sequence come. And with shotgun sequencing, there is no annotation. Those are just short reads coming from everywhere. As a result, you have this challenge, computational challenge of assembly and alignment. And as the computer technologies and algorithms started evolving, that shotgun approach became cheaper and more accessible for a variety of different use cases. Sanger technology has been continuously used even since then, and it has improved and evolved over time as well. There are some other types of technologies out there, like I mentioned, Oxford Nanopore, PacBio, etc. So there's still a variety of technologies. They have their own benefits and their own applications, and there are different costs as well. Okay, so let's see, drugs, prognosis, mortality difference. Yes, exactly. So now you're talking about different phenotypes. And so we'll talk about different phenotypes according and try to relate the phenotypic information with genomic information. Okay, because I have observed in one of the databases, there were different size of COVID patients. I'm not sure what size you mean. There are different numbers of COVID positive patients, yes, in different places. Could it mean that the virus strain alters according to the specific genomes with respect to geographic location of the host? Yeah, I think that's exactly what we're seeing, right? So the virus is evolving. We need to understand what impact those different mutations might have, but there's a number of publications out there that we will review in this program where we see the specific mutations as they occur and try to understand what they might mean. SNPs in the viral genome pools taken into consideration during vaccine design. Of course, yes. As you design a vaccine, you're trying to train the immune system to recognize a specific sequence that is not going to be at the nucleotide level. That sequence is going to have a 3D structure. And so those changes at the nucleotide level will have an impact sometimes on the 3D structure. So that will have an impact on the vaccine design as well. Can we say that the SARS-CoV-2 genotype is highly geographically spread based on variation in strains? Well, it's definitely different between different geolocations, but not all geolocations are different. So as you go back to that next strain site, try to start thinking about the different locations and the different genomes in those locations. Even visually, you can see it. And we'll actually do a comparison in this program in one of the future sessions, where we take different locations and we start comparing them. Okay, what is a SAM or a BAM file? So as we mentioned before, this is an example of a SAM or a BAM file, just so that you have a visual. And this is an actual BAM file. Okay, you can see that this is a number of reads aligned to a portion of the reference genome. And eventually it will look like this. So you see here, for example, different reads, their quality information, where they are located, etc. Visually, it will look something like this. This is also a BAM file. BAM and SAM is the exact same information in different types of ASCII code or binary code. So different types of storage techniques, but for the same information. Read and their alignment position on the reference genome. Okay, for SARS-CoV-2 sequencing, what kind of sequencing is done, like metagenomics or RNA sequencing? So it's an RNA virus, but it could be, so there are different techniques, like we mentioned. We'll talk more specifically about examples, specific examples, because different use cases will have different examples, right? So let's look at that in just a minute here. De novo sequencing of viruses, of course, that's one of the primary objectives of next generation sequencing, is to understand not only presence of a specific known virus, but also understand presence of novel viruses. So there are different ways of doing that. If we target the conserved sequence of the drug target, it will work or not for different SARS-CoV-2 strains. So I think that's a question that requires a lot of time to answer, because a drug target obviously is dependent on many different things. It's not something that is just based on the sequence. And so when we talk about targets for drugs and finding those conserved regions, we need to start looking at the variation, then understand which areas is that variation present in. And then once we have high confidence that that position is of interest and is indeed a drug target, we can then continue to sequence and look for those positions. That is something that people do. In RNA viruses, there's a lot of random mutations that will occur. So we'll talk about the correlation between sequence and structure and the different intervention techniques also in the context of the program, but in later sessions. All right, great. Thank you. And thanks for the wonderful discussion as we continue to look at this. So let's move on. In one of our first examples that we want to do and we'll do this more later, we'll discuss how different types of data that we just discussed could be analyzed in different sections of the platform that you all have access to. So as we move into this program, I would recommend that all of you try and check that not only you have access to the specific course material and project material that we already mentioned, but you also have access to this platform. So let's take a look at what this platform looks like and what can you find there. So this is the platform, server.t-bio.info. As you completed registration, you should have gotten an email about this access, about your account here. So you should have gotten, it's going to be your email address as the login, and you should have gotten an email of your password. So please, as we are now in the session, check that you can go to this link and you have your profile. For example, mine is Ilya@pine.bio. So please let me know, maybe you can type in yes if you do have access to the platform and you can see it. Okay. All right, great. Thank you, Shweta and Kita. Okay. Harish, Manish. All right, great. Renee. Okay, Marcelino. All right, excellent. So the first thing that we can do here is you will see these little arrows, and the arrows provide you with some demos. Okay, you can go through different sections and you can see some demos. If you click on a demo, okay, you'll see some examples that we will be going through in this program, explaining step by step what each one of these steps means and how it relates to the study of SARS-CoV-2 genomic data. Okay, so you can start playing around with some of these, and we will be showing you also some examples of specific examples of sequences and data files that you can take and use some of these tools. Okay, previously somebody asked, how are these tools different from doing this analysis on your computer or on some server? No different at all, apart from it organizes the analysis into a logical step-by-step process. So you'll see here explanation of different steps, what these steps are designed to do. You will also get reference information. How can you understand each algorithm that you are using? And also it allows you to process big data not on your laptop, right? So if you wanted to, for example, take samples directly from patients, and FASTQ files from those patients would be used here on this platform, it might take you, hours, maybe minutes, hours, or days, but it's not on your computer. It's accessible through the browser. If you are doing this on your computer, I know personally I've tried on my laptop processing FASTQ files. First of all, it will take days, and second of all, there's nothing else that you can do on your computer. So it's really an efficient way for you to conduct this type of analysis. In the context of the program, we also are able to help you along the way. So for anyone that runs an analysis, we can actually look at the types of analysis that you are doing, and we can start checking for any kinds of issues. And those issues can be addressed, and we can tell you about the files that you have uploaded. For example, if something was wrong there, or if some setting was wrong, and things like that. So really, this is a way for us to interact with you, knowing what's going on. Once you are done, as I mentioned, a lot of these methods are linked, so you can find that method, you can download it, and then you can install it on your computer if that is what you would like to do. Okay, so that's important. If you have not gotten that login information and you have not started looking at the platform, please start doing that and make sure that you are able to log in, or if you don't, then reach out to us. Okay, so another important thing for you to be able to do in the context of this program is start looking on different resources, online repositories of different viral genomes. Okay, so Jupiter is asking to show the demo. So again, yes, I'll be happy to show that. Again, we'll go through a lot of these again many times. There are many different areas here that we're going to be using. This is an area called virus quasi-species, allows you to really work with different types of viral data. There's also a section here called phylogenetic tree. We'll be using that. Also, we will be using some of these methods right here for metagenomics. And so make sure that you take a look. There's quite a number of different areas that we will be using, okay? Okay, if you do not have access to the platform and you have completed your registration, please reach out to Beepsha, which is marketing@pine.bio, okay? All right, what's the repeatome? Okay, repeats, yes. So let's try to, we're already out of time. So let's go on and, you know, just wanted to kind of prepare you for the next session. We want to start exploring different databases, and we will use these databases, for example, in trying to align reads, right? And this is one of the questions that we already posed here. You can take a sample from a patient and map those reads that have not been mapped to the host, and map those reads onto a database of viral genomes, and build a pipeline to annotate the presence of specific viruses, right? So to understand this process, we have an example project. This example project is something that you can learn from. And as we go through the program, you will see how in different parts of the program, we will offer you to go through some relevant materials. We'll really give you a deeper insight into how this type of analysis is performed. Now, beyond that, we will also be doing some literature review. And the links to publications are going to be present in a PDF format, so you don't have to go through any payment gateway or anything like that. You'll have access to those publications. And here we've prepared at least 3 different publications that are related to some of the questions that you have been posing here in the session. For example, there's a review on viral metagenomics, kind of gives you a history of viral metagenomics. Application of next generation sequencing technologies in virology. Again, a review of how the different next generation sequencing technologies are used in virology. And specifically, in terms of COVID-19, how can these types of genomic studies help us understand the epidemiology, evolution, and how the different aspects of virology interact to help us understand what's going on. So yeah, so these are going to be posted on the forum. Remember, Mohit explained that the program has a forum. So let's go and find this forum. We have 2 separate portals for those of you joining through Louisiana Biomedical Network, and for those of you joining online, separate from Louisiana Biomedical Research Network, right? So those are 2 different portals. If you are from Louisiana, any of the campuses in Louisiana, you will be going to the LBRN site. If you are not, and you have joined separately online, you'll be able to see that here. So here, the second tab is the forum. And it's important just also, you will get an email notification if you've started looking at that and completed your registration. And here we will have session files and resources, session recordings, and other types of questions. Any kinds of questions that you will see, you will also get answers to those questions here. So for our next time, you will see the recording of this session, and you will get the links to those publications. And if we use any kind of data sets in our session, we'll also share those data sets here. All right, excellent. Yes, and you can post queries on the forum as well. All right, so assignment for next time, start looking at some of these online materials that we shared with you. There's a project. This project explains how host reads from sequencing of the host can help us identify specific viruses. So if you would like to look into that, there's a project on that. There's a blog post on the different virology and genomics resources. You can look at those. And there's also a course on introduction to metagenomics where some of this metagenomic sequencing is going to be explained in greater detail. Okay, so this program, I'm just going to also briefly give you an overview of the program schedule and the different sessions that we'll have and what we'll learn in each session. So in today's session, we talked about sequencing and how sequencing is being used to study viral genomes. Then we'll start talking about finding specific viral genomes in animals and patients using next generation sequencing. So how do we actually take a sample from a bat or a pangolin or a human and identify what kind of a virus is inside? Then we'll talk about different types of databases for viral sequences, where we'll continue the same analysis, but we'll start looking at good and bad examples and what kind of information can we extract from these different databases. And then we'll talk about sequence similarity, so multiple sequence alignment. How do we study similarity? How do we also study differences? And how do we develop consensus genomes, or what is the prevalent genome in a specific sample? Then we'll talk about finding specific data sets from different locations and different types of sources, like different hosts, different locations around the world, different time points where those were sampled, and using those for analysis. And we'll do both exploratory analysis, where we just kind of get an overview of what's going on, and then developing a specific analysis question. What do you want to have answered with this type of data? Then we'll talk about phylogenetic analysis. How can we compare sequences and understand their relationships? How are they related to each other? Or how are they different and potentially have common origins? So that's called the phylogenetic analysis. Then we'll have a hands-on session where we'll take those concepts, where to find data, how to compare the different data, and what pipelines to run. And then we'll talk about the different sections where you can do different types of analyses. We have these different sections that we've prepared that you can try and play around with, and they will provide you visual and specific outputs that you can learn from. So in the context of this program, we'll go through the different materials that we discussed today in the different online courses. We'll do some practical analysis as we've discussed, and you'll also have these interactive sessions where we hope that you are able to interact with us and really ask any questions that come along the way. And then we'll go into the application, right? We'll talk a little bit about some of those applications of these kinds of studies for vaccine design, for drug design. So what is the relationship between the sequence and the structure, and how do we use that relationship to understand the impact on preparing the immune system to identify COVID-19, as well as trying to disrupt some of the processes that it leverages to replicate and go between different cells as an antiviral strategy. Okay, so this is the overview of the program. Again, any questions between now and then, please post them on the forum, right? Again, thank you, Renee, and all of you for joining today. We hope to see you again on Friday. If you want to have something specific answered by next time, we'll either answer it on the forum or we'll cover it in the next session. Excellent. Thank you very much, everyone, for joining today. The recording will be available. And thank you, Mohit, for starting off this session and explaining the registration process. If anyone has any questions about how to complete the registration or get access to any of these resources, please make sure to email us. Thank you very much, and we'll see you again next time.